of the afternoon. Next up, we have Brittany Scannell visiting us from Stony Brook.
Hello, so my name is Brittany. I am a PhD student at Stony Brook University. And so this is actually our first time coming and presenting at an ACT meeting today. So I wanted to give you guys kind of a broader idea of what we're doing on Long Island and a little bit of an exploratory analysis of kind of what our first data looks like. And so I'm part of the Peterson Community Ecology Lab up on Long Island. And the two main projects that we have right now looking at acoustic telemetry are our wind farm export cable and our New York State DEC conservation artificial reef projects. So for these two projects, we have had a massive tagging effort over the last 2 years. We have tagged 13 different species with 6 different teleosts, 3 different types of skates, and 4 different types of sharks, which is amounted to 457 individuals over the next 2 years. And between those 2 projects, that's something like we have 800 more tags to deploy in the next 3 years. So we have a tall task ahead of us, and all those tags will be going out on these species. So there are a couple of species out of those 13 that we're really focused on. The New York State DEC is focused on things like reef-associated fish, which are black sea bass and tautog. So with the infrastructure of the wind farms and the artificial reefs, we've been able to tag 59 black sea bass and 20 tog. And we're also interested at the habitat use of dusky sharks, sand tigers, sandbars, and smooth dogfish on the artificial reefs. So we've so far tagged 25 of the dusky, sand tigers, and smooth dogfish, and 12 of the sandbars. So a little bit more about the artificial reefs that we have off the south shore of Long Island. New York State got into the artificial reef game quite early in the 1920s. They've had a dedicated program expanding and adding this hard bottom structure in an area that's naturally soft. So these artificial reefs, they enhance the environment by creating these biological hotspots that provide food, they provide shelter, they provide hard substrate for things like mussels, algae, and barnacles to settle on. And then we have fish like the black sea bass and the tog that come and they use it to forage and shelter and as spawning grounds. So New York State has an array of 12 along the south shore, 2 on the north shore, and 2 inside the bays. Each of the artificial reef areas are composed of patch reef. So what the DEC does is they'll take cleaned and recycled materials and they'll place it at discrete areas within inside of the reef, creating patch reefs. So this allows some of that natural soft bottom to exist in between the areas, actually increasing diversity and production within the site. And if we zoom into what one of the sites look like, we have all different material. So we have things like bridge structure, rock, vessels, armored tanks. And so far what we've had is receivers that have been moored on the structure or inside of the structure matrix in box patterns. So from these box patterns that we've had at our initial artificial reefs, we've been looking at residency patterns of black sea bass and tog. So specifically looking at tog, we've seen that we only see them at artificial reefs that we release them at. So if we tag a tog at Shinnecock Reef, it only stays at Shinnecock Reef. So there's no movement for those species in between different reef areas. For black sea bass, it's similar. So they're not actually switching between artificial reefs. We have 12-mile reefs with one of our black sea bass visited, and that visited on its way out. So we can see black sea bass will migrate offshore in the winter. So in the summer and the fall, it's at the Shinnecock Artificial Reef, then it moves off to 12-mile, which is even further offshore. And then it returns to the same exact reef area that following summer. We've also been interested in how larger transient predators are using the artificial reefs. So here on the left, we have kind of what the water column would look like. We have the sand, and we have structure that at the Moriches Artificial Reef comes up to about 18 meters. And then at the surface in the summer, we have bait fish like Manhattan that are swimming up and down along the coast of Long Island. And if we zoom in to the Moriches Artificial Reef and look at 2 of our shark species, we have duskies and we have sand tigers. We can kind of start to see how each of the species differently use the water column. So unsurprisingly, Duskies are mid-water opportunistic feeders. They spend a lot more time at the surface, and we also see them moving along the coast a lot more than a fish, a shark like a sand tiger, which spends far more of its time actually at the depth of the artificial reef structures, where it's kind of waiting more of a predator that's sitting on the bottom, only coming up occasionally to gulp air. And we've started pairing some of our acoustic telemetry with, we were taking stable isotope blood work, and we're taking cloacal swabs to look at relative diet indexes on some of these sharks to possibly try to pair it to some of the fish that are on the artificial reef. So we did a sample of 14 Duskies this summer, kind of as a pilot study, and we found that their diet mainly composed of 3 species. So unsurprisingly, most of their diet is bunker. Then one of the Dusky sharks had bluefish in their diet, and one of them had lots of sea robin, which in and of itself is a benthic fish. So looking more at our future goals, we were looking to use a more fine scale approach to determine what type of structure pieces these fish are spending their time on, where inside the artificial reef, what depths they're using. So starting this summer, we deployed VPS arrays at all of our artificial reefs. And a great example of that is the Shinnecock Artificial Reef. So we have a series of over 20 receivers that cover 25 acres of reef that we now have in a fine scale positioning array. And our second major project is the submarine cable project for the wind farms. So over 2 different projects, you can see here is a map of the wind area and some of the proposed cable laying avenues. Those 2 pins are the 2 sites that we monitor. And so basically what these submarine cables do is they transport the power that's generated at the wind farm sites. And as the power is running from offshore to onshore in that cable, it produces EMF. And so this project is a before, during, and after construction project to look and see how that EMF is going to affect the fish and invertebrates that are in that area. So we'll be looking at things like striped bass to see how it's affecting fish that are higher up in the water column, skates and horseshoe crabs to see things that are right on top of those benthos on the cable. And that has kind of culminated in a receiver array for us that stretches all the way from Western Long Island out to the east end on Long Island, 2 receivers on the North Shore and 5 in Peconic Bay, all looking at different types of species and at different scales. And so on Long Island itself, this is only probably half of the receivers that we have out. There's another lab that we work with that kind of has their own, I think they have 70 receiver arrays, the Frisk lab, that we share detections in between. So between the 2 of us, we cover a lot of area on Long Island and hopefully are capturing your fish as they're moving up and down the shore.
Does anyone have any questions for Brittany?
I would just say that there's enough of us doing this now that it really deserves its very own symposium, where people get together and work on a shared coast-wide cable crossing question, how to analyze that and put that together from all the shared partners, because we're doing the same thing. Our fish are going up there, and I know we got a lot more detections from fish than we put out than from fish that we put out. So yours are coming through there, and it's worth a symposium. We should start putting something on the calendar.
Totally agree.
Or a workshop, and then it's.
But the issue of analysis, no.
Yeah, it is.
Yeah, to kind of
Right.
standardize it between
Right,
everyone.
exactly. It's worth its own very own workshop.
Let's
So,
do it.
comments.
So, I mean,
Yeah, so if people couldn't hear that online, there was just a brief discussion that there's so much work going on related to offshore wind and in that same general area that it warrants its own meeting and workshop to think about how to approach analyzing the data in a shared way and thinking about it as a broader whole.
Yeah, so for the swabs, it's DNA metabolic living. And then it's looking at NU fish primers and S16 and
Anybody else? All right, next up, we have Thomas Grothus from Rutgers.
Thank you.
You should just be able to use the arrow keys.
All right, so this is a little bit of a departure from the shared network theme, but by invitation or request, a synthesis of work that we've been doing going back to about 2009 on the use of AUV. So I want to stress that the AUV is different than an ROV. It's truly robotic. Oops. So it's not controlled. It's actually capable of sensing the environment and sensing data and then making its own decisions about where to go after that, although it's base programmed to follow a path. And it is different than a glider. I'll compare that a little bit. Oops. The niche of the propelled ROV, and here you see the propeller on the bottom of that one, really is for places that you can't put a ship, but different than a glider and some other things. Again, it can make decisions. So frequently on a boat, you'll go out and you'll look on a broad scale and track something, or go to find an animal that's tagged, find it, and once you detect the tag, you'll make a local maneuver to sense something about the environment that tag is in, and in fact, nail down the exact position of that tag potentially to meter scale. So that's one of the major differences between the AUV and the glider. So glider is an endurance tool, and it's very good for scoping for long time or very wide areas. The AUV can only do that on the order of a day or so on 35 kilometers, but within that, it could search 35 kilometers, maybe a kilometer to each side based on tag range, but then make decisions and really nail that down to potentially sub-meter precision, the location of that tag. The pedigree, and I'll go back one slide here, is, oops, the side scan, jumping around too much here. You see the side scan rail on the side of that, and the pedigree of that vehicle is a minesweeper. So it has that side scan on it, and you can use that to couple tag detections with not only the environment, but also even individuals. So in this case, you have a whole bunch of short-nosed sturgeons up near Trenton. They're staging just before spawning after ice out. You can see that's a drag mark from a buoy that was caught in ice, and you can see the kind of the sand rays, sand ripples, and some of these were carrying tags, and because the AUV is also detecting those, that corroborates it. Bigger animals, you can actually see individual, like the manta ray there, different stingrays, angel sharks, you can tell them species. And I've seen those, the aerial photo that Mariah showed of all those cow-nose rays. We've seen that underwater on the side scan as well. So that's where it really gets put together. But there's also a niche in this in a tag difference. So there's tags that perform particularly well with what the AUV can do. And that will become apparent a little bit later. But in general, these are the kinds of things that need work, that we have been working on when you consider AUVs. So the tag is a number of degrees of freedom, right? The code, the rate, the range, the precision. The AUV itself, what kind of AUV it is, because there's many different kinds that have different speed, depth, and mount constraints. They all affect the sensitivity, right? The mission, how you program the mission of the AUV, how you deploy it, the mission paths that it swims in. And so from any of the kind of intersections of these different things, you get presence and absence, but you can really hone that down to efficiency and precision in position when you consider all of these different things. And that comes from an approach that uses both modeling, because they're expensive to just stick out, right? So you can do a lot of this thing in simulation and change these parameters, and then you can test them in the field. And that's what we've been doing. So a couple of these different degrees of freedom. Here's a coaxial mount of a low-tech hydrophone on a torpedo-shaped AUV. Here is a parasitic mount of, by the way, the low-tech one that you saw previously is integrated with data flow to the vehicle's navigation. This is simple limpet attachment of a VR2. We continue to use this, but now we're actually using the much smaller VTs, less drag, and they can ping, so you can actually use them for standardizing. And then here's a different orientation, and this matters because there's essentially a toroidal efficiency around, like a donut around how these things are situated. So those are the kinds of things you want to test. In depth testing, we ran the Remus with a low-tech hydrophone on it through a field of transmitters, test transmitters that were moored. This is up in Alaska, in the deep passages, with tags down at 500 meters, 200 meters, 100 meters, and 20 meters on these deep arrays. And the AUV alone detected, so we ran the AUV and we ran the boat right on top of it, so they followed each other on the same path. And the AUV detected the tags down at 500 meters, and both the boat and the AUV detected the tags at 200 meters, but the AUV was much better at that. Then at 100 meters, the boat and the AUV are about equal. And then at 20 meters of really shallow tags, the AUV did better again. And I think that's because you have stratification, especially in Alaskan waters, where you can have a high density stratification, and it's the angle of the tag coming up. The AUV is swimming down at about 80 meters, and it's doing better. And part of that is the AUV is in a quieter environment. We tried this again with hydrophones to get the towed hydrophones on the boat to get them down, and then you suffer line loss, because to get the hydrophone on a tow line down from a boat that's on the surface, you're getting down 200 meters of cable or something to get it down that deep. So one of the things is that the signal's coming up as kind of a bubble, and at 500 meters, that's at the surface where the ship is. So the tangent is the surface, and there's kind of one place where you can intercept that. But if you get the AUV down at 100 meters and run it through that hemisphere of reception zone, you're already taking a chord through that hemisphere that's just much, much longer. So the concern is that the noise of the self noise of the AUV may occlude some of the tags. We've looked at that. What the first graph shows is that that regardless of the bearing, so you're detecting tags in all directions, including those behind the vehicle, in front of the vehicle, and to either side, but especially get the far away, the detections from reference transmitters that are quite far away. You're getting those primarily when they're broadside to the vehicle. And again, that's that reception donut in the coaxial ones. It's hearing those furthest away. And then as far as the power of reception, when you're looking at this recorded signal strength, you can see that that's basically a square plot, and it doesn't matter where they are. If they're close, you're going to hear them loud. If they're quiet, you won't. So simply mapping them is one approach. This shows a path where we're getting Femco detections going up there, going up the river, and you're just looking at the timing of the vehicle. The highest detection, or the most number of detections, are going to be where you're closest to it, if those detections aren't too far apart, right? If they're not like 2 minutes. If you're getting a tag that's pinging frequently, you're going to get a real cluster that you can smooth, and you can isolate the position of that a little bit better. But we can do better if we're using tags that do not have invariant timing. So the Vemco tags have variant timing because of code collision, but low-tech tags and some other tags do not have that. So you can have them pinging 2 seconds or 5 seconds or 6 seconds, whatever you program exactly. And you could have 50 of those in a bucket going at the same time, and you won't have code collision. So that means that you can do a synthetic aperture for that with that. And what that essentially is, it's very similar to Doppler shift. And as you're going away, the time gets longer. So basically, imagine you're moving forward and you hear a tag and it's pinging at a 5-second interval. You expect to hear the next ping exactly 5 seconds later. But if you hear it 4.988 seconds later, you've gotten closer to the tag. And until you pass that tag, it keeps getting shorter until you pass it, and then it starts getting longer. And now you have a bimodal resolution. You know exactly where that tag is that you've passed. As soon as you hook a right or a left, you can resolve that bimodality. If it's shorter again to the right, it's on the right side, and you throw away the left side detection. This is very similar to what happens in a virtual positioning array. You're using that same math. You're just taking and dropping off essentially a bunch of hydrophones along a path, and then you're resetting the timing of those depending on the elapsed timing of the vehicle as it's passed. So in order to do that, you want to have payload control, which is to say that you have a pre-programmed route, versus something that happens once you hear a transmitter. The way we do that is the receiver talks to a guest onboard stack on the vehicle. So the telemetry hears that, it sends it to the little software package that we wrote in C++ called Sailver. It goes through a decision tree. If it decides it should do a maneuver, it passes that to the native navigation stack of the AUV, and that sends commands to the control to turn left, right, go fast, slow down, go up, down, whatever you want it to do that forms a good aperture. So basically this is what it looks like. It's coming along. It hears a tag. Okay, and we've been using this particular maneuver. You can make those maneuvers big or small. And you're going to make that decision based on what it is you think you're tracking. So you can actually look up on this if the tag you're looking, so it detects a tag and it compares that to an onboard library. It says, okay, this tag I know belongs to a blue crab or a ray or a shark, and it's going to assign a different maneuver to that animal based on what you think that animal is likely to do, right? A crab is moving very slowly compared to a shark. And so you're going to use a bigger array or the strength of the tag for that matter. So all those kinds of things have now been tested, including delaying it. We found that if you hook too early, you're hearing that tag from far away and you're not making the maneuver right where the tag is, but before. So delay it and hear it later and you get better results. But this is basically the flow chart. You're looking at, is this a new contact? If it is, I may act on it. If it's one I've already acted on, I don't want to do that again because I'll get stuck in a feedback loop. So you put it in a cool down and then pick a template and pass that on to the override computer on the, and we've added these kinds of things also for safety. We put the AV into the water and right away it detected a fish and started a maneuver, and the boat wasn't out of the way yet. So we put a delay in that. We put delays in for safe areas, like if you're in this area, don't execute a maneuver. And we've checked those on arrays in the water. Move on here. What happened? I touched something. And we found that there's really good repeatability. So if you have test tags in the water and you run this mission, the response maneuvers happen reliably in the same place that they did over and over. And also when you have them detecting a tag in an area where they're not supposed to and they hit that override block, it gets blocked and it's not executing a maneuver. There's a number of different solutions that can be calculated from a collection. So all these stars represent where you've detected a tag along the way. Use those in a particle distribution function that solves for where the actual tag is. And it gives you many different results, many of which might coincide, but other ones are far away. Those can be screened. There's a number of different ways of assessing those, basically dilution of precision, which is the geometry of the ray that you've calculated. That shows up as a byproduct of the calculations, same stuff that gets used in GPS. And you can throw out a bunch of your solutions and look for ones that have a very good dilution of precision and a very, very low residual. And we've mapped those against known arrays of test tags. So that's what you would call a agnostic metric. So we know where the tag is and then compare that to the data set where we hold back that. So here, for instance, you can see the relative signal strength versus distance from the hydrophone. And we know that by about 750 meters, we're not hearing any more tags anymore at any level. And so out of all these, a bunch of that's junk and you could filter those. And then you take the rest of them and you look for residual and the distance that it is from it. So actually, in this case, you're seeing that none and large, all of them, all different kinds of maneuvers. Just as an example, we go into deep depth with this. There's lots of solutions that you throw out. The ones that are really far away typically do not have very good resolution, but you can get down to meter scale with these. And part of that is the scale at which the AUV's position itself. Doing it by residual, which seems to be the best one. You can see the small maneuver worked especially well. I think that's because there's less time in which the aperture is formed. So just to finish out then, these are the needs that we still want to do. We still have payload control, model of the correct maneuver given fish speed, sound pressure level, signal repeat rate, time since last maneuver, because again, you don't want to get caught in a feedback loop. So you put on a cool down, but then you could potentially miss this. One of the niches here is for repeated measures. So we tested this on dispersal of salmon smolts from their head river into an estuary in Alaska to see. So we're running the AUV every day back and forth, and mapping the cloud dispersal of these. And that's very different than using this to track actual fish, which has been done with this kind of system, but it's a very different application. So that's a Lagrangian versus an Eulerian approach. These are things that we need to continue working on. And then questions. Did I go over, way over? Okay, cool.
And can you answer them from up here too? I'm gonna work on switching it out.
Yeah.
Give a couple more practical applications for.
Yeah. So right now, we're using this actually to look at response of fish to borrow pit or to sand dredging. So we want a large scale approach that we have hydrophones down, moored hydrophones that are looking at everybody's fishes that move by there when they do it seasonally. This is before after control impact design. And then in very fine scale, even finer than just a 2-minute trawl can show you, you can see where the fish are orienting. The borrow pit in this case is not a pit yet, it's actually a ridge, an elevated ridge. And we're finding from the trawls already that there is very fine scale differentiation between the lower side of the ridge, the other side of the ridge, the top of the ridge, and then the control areas. And so we want to do that with meter scale resolution in terms of tracking as well. So we can have the AUV going out there. It maps the bottom, so it sees the changes in the sediment, both spatially and temporally. In the side scan, it's mapping pelagic fish like herrings and stuff like that in the water column from the side scan also. And then finally, it's measuring where particular fish spend time. So most of the, for instance, ocean trawls, survey trawls, they trawl over 2 kilometer area. in the stock assessment, and we're finding that that already integrates way too big of habitat in sand. You think sand is kind of uniform, but on these ridge features, it's not. And even very short 2-minute trawls in these areas were very different by assemblage. And so we expect to see that in movement response too.
Thanks.
Any other questions? I should have repeated that question, actually. Hope it was got.
All right, so our last, but very exciting talk will be from Lacey Huey, who works at our National Zoo and Conservation Biology Center. So she gets to work with all kinds of things, not just marine life.
Yeah, as Kim prefaced, I actually come from the terrestrial tracking world, so this has been a really enlightening, here? Oh, okay. Enlightening and helpful meeting for me, just to speak to you guys. Part of my role is to, identify synergies between multiple tracking methods and ecosystems. So got all kinds of ideas from this meeting. So thanks for inviting me and for this opportunity to just talk a little bit more about movement of life. So that's Smithsonian's pan-institutional initiative to understand how animals move across changing land and seascapes. And I don't need to convince this crowd that movement is essential for life on Earth. And but I just like to spend a moment talking about exactly why we care so much about movement. And it's because it underpins these really basic ecological and biological functions. So it connects ecosystems, it drives key biological processes, and underpins healthy communities of humans and wildlife. However, we still know very little about the impact of a changing planet on the movement of species. So that's why what we all do here. And Smithsonian, actually, this is a bit of a side project that I've been working on with some of our scientists here, learning more about the history of animal tracking. And through that process, we learned that actually Smithsonian has been tracking animals for about 100 years and had some of the first scientists to band birds. So Paul Bart here. And there's some really interesting history about Smithsonian's role in the Pacific Ocean Biological Survey, which I'm sure many of you know more about than I do, but I'm just learning about it. There was lots of controversy around that project because it was with Department of Defense and ended up, I think still it's unclear, but there were some suggestions of ties to biological warfare. And so through that process, that Smithsonian scientists were banding millions of birds, so almost 2 million birds were banded in that study. And an interesting kind of also side note to this story is that the Gemini 9 space shuttle mission, when the crew, there's a ship out there tracking it to recover the astronauts, and I guess that the crew was fishing, longline fishing for their dinner. And they actually ended up catching an albatross on the long line that had one of these Smithsonian tags on it. So this is an endlessly interesting story. We're still researching it with Smithsonian historians and coming up with some ways to tell the story of animal tracking, both through the lens of history and culture and science. So I'll talk more about that in a little bit, but I want this to be a community effort. So if any of you have cool historic animal tracking stories or photos, I'd love to talk with you more. I'll have a more structured outreach campaign for this in the next few months, but just flagging that for anyone that wants to talk about it. So from that work, we've really grown into a globally distributed network of animal tracking projects. So like I mentioned, we have scientists studying terrestrial species, marine and aquatic species, and a large, probably our longest running research program at the Smithsonian Migratory Bird Center, studying birds. So this is not even a comprehensive map, but our best map of projects over time. And our team, so Matt Ogburn here is our lead for the marine systems, and Chris Fleming is a quantitative ecologist. He does a lot of work with home range estimation, the CTMM package, if anyone, I know it's more in the satellite tracking world, but he's developed that package. Autumn Lynn Harrison at the Migratory Bird Center. I'm the program manager. Peter Leingruber leads the Conservation Ecology Center at the Conservation Biology Institute. And then Mel Songer and Jared Staubach are also terrestrial leads on the projects there. We'll talk a little bit more about all of our work. So our approach really centers on 3 pillars. So doing basic and applied research, working closely with stakeholders, making sure that our science is both informed by community needs and serving community needs, and then a strong emphasis on outreach and education. So some of our basic questions we're trying to answer, a lot of which overlap with what you guys have presented here. So just how much space do animals need to survive? How are barriers influencing access to critical habitat? And how will predicted changes in movement affect socio-ecological systems? And then we do a bit of methodological advancement, so both quantitative and in the hardware realm. And one of the, I think, kind of most fun ones to talk about right now is linking physiological metrics of health to animal movement. And one way that we can do that is by working with the animal collections that we have, both at the zoo and at the Conservation Biology Institute in Front Royal. And this is a photo of a maned wolf. It's a it's actually a frugivore native to Brazil. And we have a few in captivity in Front Royal. If anyone would really like to come see them, you're welcome. We'll give you a tour, let me know. And so we have a partnership with Medtronic, which is actually taking heart rate monitors designed for humans and implanting them into maned wolves. We have maned wolf, jaguar, and discussions of elephant, but I don't think we've done anything with elephant yet. But they've been tested in the captive animals and are this year going to be releasing our first cohorts of reintroduced jaguars into the wild with these tags. We also do quite a bit of animal reintroductions. So building off of some of the species that we are raising in captivity, but actually trying to contribute and establish populations in the wild. So we have scimitar horned oryx here in Chad and the Guam kingfisher here, where it's given some really great opportunities to understand more about just the basics of reintroduction ecology. So what's happening to these animals that are being reintroduced, but also getting into some of the social interactions between animals. So at least in scimitar horned oryx, every animal that's released gets a collar. So there's some really exciting research coming out about how those groups are interacting with each other on the landscape, and also with groups that have already been established in the wild. So working with stakeholders, we've really tried to make sure all of our work is grounded in a conservation need that's informed by the communities where we work. So I'm sure many of you know Hector Guzman, work at the Smithsonian Tropical Research Institute with whales and reducing ship strikes, defining areas that can limit ship traffic outside of whale high use areas. There's a lot of work on anti-poaching in Myanmar with elephants. And then stewarding big data for conservation. So I'd say Atlantic Cooperative Telemetry Network and Kim and Matt's work with, stewarding this data for beyond the Smithsonian community is something that we really want to support. There's the new Shorebird Science and Conservation Collective, which was just formed last year as part of under the National Zoo. And they're built from the ground up integration with agencies and NGOs and communities is inherent in that model to try and get data to make the most use of each other's data as possible. And being Smithsonian, education, public outreach is probably one of our greatest strengths. We have many amazing outlets for the work that we do. And we also have a strong educational component. So to our movement work, we're very involved with Animove courses and interested in getting more focus on the quantitative challenges associated with marine and aquatic tracking. So that's something that we are trying to push on. We have the Smithsonian Mason School for Conservation, which is a physical campus, satellite campus from George Mason University that's at the Conservation Biology Institute in Front Royal. So we have undergraduates in conservation and veterinary studies that spend semesters out there and work with Smithsonian scientists. And then we give many guest lectures throughout the year. We have paid internships along the way, and but both undergraduate and graduate levels. And we emphasize field and lab-based training, and then also fellowships. So the Smithsonian Fellowship Proposal, Smithsonian Institution Fellowship Proposal, yeah, program is one avenue for graduate students, postdocs, and even, I forget the name of the exact, what is it? Senior researchers to, like, if someone's on a sabbatical, you can apply and come and stay with any Smithsonian scientist. So Matt and I both always are trying to recruit folks for that. So talk to us if that's of interest. And then we rely heavily on university partnerships, as many of you here are from our partner universities. And then something that we're trying to really build on is integration with the museums and the zoo. So we had to get the story of animal movement and animal movement science out to a larger community, not just scientists. So last year we had a lecture series called Women on the Move. That it's online now. I can share the link to that if it's of interest. It featured female scientists, including Kim. Yeah, Kim gave a great talk on that. So I encourage you to check it out. This picture on top is the new birdhouse that's opening at the National Zoo this spring. The exact date hasn't been announced yet, but it's going to be a beautiful space, physical space with interactive exhibits featuring bird tracks. And actually, I think I just heard there's scientists on display doing their work at the computer. So yeah, you can check that out. And then the One World Connected exhibit at the National Air and Space Museum. So for the satellite-based tracking projects, there are some transmitters in there, and we're actively working with that team to develop some live events, virtual events, and a traveling exhibit that will also hopefully be an outlet to tell the story of the history of animal tracking, both at Smithsonian and beyond. And a sub-project of that project is this podcast that's coming out February 23rd, where we're talking about the first elk to be tracked from satellites. Her name's Monique. If you're unfamiliar with it, I encourage you to Google this story or listen to the podcast. It's really, you can tell it's a complicated, interesting story of technology and ethics and, you know, the kind of how and why we track animals and where we've come from and where we're trying to go with this. So that's a fun one to check out for, even if you don't, if you have folks in your life that aren't excited about animals for some reason, but maybe excited about the technology, that this podcast is really for space enthusiasts. So I'm happy to take questions.